So today we're going to discuss paper 2 for your trial AS trial exam. So figure 1.1 is an electron micrograph of part of a eukaryotic cell. Okay, so question A asks the student, state how it is possible to deduct that figure 1.1 is a transmission electron micrograph. Transmission means the light will actually, the electrons will pass through the specimen and then shine onto and hit onto the screen. Okay, so transmission electron micrograph and not a scanning electron micrograph. So SEM, scanning means you only look at the surface of the structure. It's like no photostat machine, you look at the surface of the structure. So they ask you for this figure, how do you know that this figure is taken from TEM rather than SEM? Okay, so very easy. If it's SEM, scanning, you will not be able to see the structure inside the cell, because when you scan the cell, you only look at the outer contour of the cell, like cell surface membrane, and then it is spherical shape, right? So that's for SEM. SEM. So for TEM is light microscope. Okay, the only difference that we're using electron rather than light. So that's TEM, transmission electron microscope. So therefore, the electron pass through the entire cell, and whichever part that's electron dense, it will appear as dark color. So you can see the structure inside the cell. So therefore, as long as you tell me what are the structure, at least one structure that you can see in figure 1.1, you will get the mark. Of course, they already label for you mitochondrion. So those of you who write mitochondrion is visible is already correct. Of course, other structures that you can see is like this is the vesicle, Golgi apparatus are visible. Okay, then you get the mark. Ribosomes are visible, the dot, because this is electron microscope, you can see ribosome, then it's correct. So 1 mark for this. Let's look at the mark scheme. Mark scheme says, idea that you can see the internal structures of the cell. For example, you name the cell structure that you can see, such as mitochondrion, or you say that you cannot see the surface contours, because scanning electron micrograph, you see the surface contours. Okay. Also, except if you see that it's not 3-dimensional, because scanning, you see 3-dimensional, whereas TEM, you see 2-dimensional, just like light microscope. AVP is, for example, refer to smaller depth of field. This one, we are not going to talk about it. Okay. So next question. Now, if you have any question, please stop me to discuss, okay, further. So identify structure A and state one structural feature of A that supports your answer in B. So what is structure A? So structure A answer is Golgi apparatus or Golgi body. A lot of students write this as rough ER or smooth ER, so it's wrong. First thing is, now this is a stack of flat membrane structure that arrange in layers, and they are not interconnected, and they are also not connected to the nucleus, and they have got vesicles at the edges. So therefore, this is a Golgi apparatus rather than rough ER. So if you want to look at the mark scheme says this is a Golgi apparatus or body or complex because it is made up of sacs that appear in layer form, or they stack on top of each other, one another, and there are no connection between the stacks of membrane-bound sacs, okay? Not connected to each other, not connected to nucleus or nuclear membrane or nuclear envelope. There are swellings at the end of the sacs, or there are vesicles at the end of the sacs, okay? So in this case here, some of the students will need to learn up certain term that is used to describe the structure, biological structure, like sex, means membrane bound bag. Okay, so those of you who have not learned up, you please learn up the word to describe structure. Okay, C is actually calculate the actual diameter of the mitochondrion and give your answer in nanometer. So that means you have to go to your paper, and then in this question already given you the magnification, which is M, right? And then you put your ruler from X to Y. So that will give you your M equals to I over A. So it will give you your I, the image size. Then you can calculate your actual size and you have to convert it into nanometers. So I think in this question, I find that a lot of students, 50% and above of students, do not know how to do conversion, which is very surprising for me. Okay, first thing you need to memorize the units. What do they mean? And second thing, you really need to know how to do conversion. And third thing is some of you lost the mark because you didn't write the formula. You didn't write the formula clearly to show your working. You didn't show, yeah, that's the main reason why students didn't get the mark for this question. Okay, can you see that first marking point is you need to indicate the formula. And I highly advise you not to use the symbol, even though I give you correct, but it doesn't mean the examiner will give you correct because I do not know what is M, I, and A unless you tell me what do you mean by M, I, and A, because they are not the official short form. Like just like physics, if it's official short form, then you can use that MIA, but then this is not official short form, so you have to spell out magnification is equals to image divided by the actual diameter, actual length. So this is 1 mark. Second mark is given for correct answer. Correct answer means as long as your calculation is correct, you will get, okay, you will get 277, 287, 298, 309, or 319 nanometers only, because it is based on this value that you have obtained from measuring. So that means, I forgot what is the value it is. Probably is 13, 13.5, 14, 40.5, and 15, I think. Okay. Oh yeah, it's actually based on this one. 14 plus minus 1. Okay, 14 plus minus 1. Yeah, correct. So it is, if it's 14 is this one, right? 13.5 is this one. 13 millimeter is this one, based on your measurement. Okay, so therefore any other answer would be wrong. Another thing is why student didn't get correct, even though calculation is correct, because the rounding up part is wrong. That means you use your calculator, calculate it when you round up. Let's say, for example, your calculator show 276.666. Then, of course, your answer must be 277 rather than 276. So again, rounding up mistake, no mark. So don't make this kind of mistake in exam. Okay, it's not worth to lose that mark, right? D. So it says that, okay, this is a very interesting question. The inner membrane of mitochondrial envelope is much less permeable than the outer membrane. So it's less permeable. Okay, but I didn't say less permeable to what. So that means to say it can be less permeable to maybe water. It can be less permeable to the ions. It can be less permeable to the, CO2, oxygen. Maybe it's less permeable to glucose, right? So these are all possibilities, right? Okay. So suggest one way in which the structure of the inner membrane of the mitochondrion differ from the outer membrane to produce a less permeable inner membrane. So like water, CO2, and oxygen, right? They pass through the spaces the spaces between the phospholipid molecules. Okay, so this is phospholipid bilayer. So these molecules that are uncharged but polar, they can pass directly across the spaces in between the phospholipid molecules. So when you say if the membrane is less permeable, that means to say this gap between the two phospholipid molecules is becoming smaller. So therefore, why is it becoming smaller? So most probably is because they are aligned more closely. So the phospholipid molecules are aligned more closely due to saturated fatty acid tail. So you know that when the tail can be saturated and unsaturated. So it is when the tail that is saturated, okay, that the neighboring fatty acid tail can align close to one another. So let's say this is one fatty acid, one phospholipid molecule, and this is the adjacent phospholipid molecule. So because the fatty acid tails are saturated, they are all closely aligned. So therefore, the gap between these two becomes very small. So those molecules have got lower chances of passing through compared to if the fatty acid tail is unsaturated, then you will see that probably this one here, okay, got unsaturated, there will be a king. A king means suddenly there is a king and it bent the other way. Okay, it bent the other way this way. Okay, sorry, to draw again. Undo. So originally it's a line, but then there is suddenly a king. Let's say a king that go this way. So it's pushed away. Okay. So there is a big gap here. So this tree can pass through more easily. So this talking about fatty acid tails are more saturated. So there's the gap between phospholipid molecule becomes smaller. Okay, so this is the best answer. Of course, second is possibly there could be a more cholesterol. There will be more cholesterol in the membrane. So when it's more cholesterol will be seated in the gap right between the phospholipid molecules. So that will also deter, block these three molecules from passing through. So that's second answer, more cholesterol. Okay. And also if you're talking about ions, right, and glucose, which requires the channel protein to transport the ions or carrier proteins to transport the glucose, then you can say that the membrane has got less channel proteins or less carrier proteins. So less channel protein, less carrier proteins. These are also correct. Okay, so these are the answers. So I think no need to see the mark scheme. Part E. So part E is talking about signaling. Okay. So let's read the main part of the question. So it says that phosphatase enzymes remove phosphate groups from organic molecules, while kinase enzymes, While kinase enzymes add phosphate groups. Okay. Read the following passage and suggest why hormones referred to the passage are unable to enter the cell. So why hormone cannot enter the cell? Some hormones circulate in the blood and are able to trigger transcription within the cell, even though they are unable to enter the cell. So cannot enter the cell. Phosphatases and kinases then take part in cell activities, eventually result in genes switching on and transcription begin. So hormone, there are two types of hormones. So here you need to learn and you need to know very well. There are two types of hormones. So I'll show you the endocrine gland slide first, okay? Okay, here. So there are two types of hormone, either non-steroid or steroid hormone. So non-steroid hormone are actually usually the protein. So protein is big, it cannot pass through the phospholipid molecule. So it will attach to a receptor for the hormone, which is found on the membrane of the target cell. Whereas hormone that's made up of steroid hormone, because steroid is part of lipid, it can pass through the membrane. So therefore, it will pass through the membrane and then bind to its receptor molecule, which is either inside the cytoplasm or inside the nucleus. So therefore, there are a few types of molecules that can form our hormone. First one is protein. So the insulin and glucagon that you've learned, they are protein. So they have to bind to the receptor on the cell surface membrane because they're too big to enter the cell. Whereas steroids hormone like these two here, estrogen, progesterone, thyroxine molecules, steroid hormones, okay, they've got the organic rings there. They can pass through the membrane and then enter the cytoplasm. The receptors are in the cytoplasm. Okay. Then we've got fatty acid. We've got derivative of amines that form the hormone like thyroxine, adrenaline. So thyroxine is derivatives of amine. So the receptors are in the cytoplasm. So therefore, when the question asks you why the hormone cannot pass through the membrane, then you know that it must be made out of protein. Okay. So therefore, the answer is they are made out of protein. Where is it? Okay. So therefore, it is too large to cross through the membrane. Or it is hydrophilic, so it cannot pass through the hydrophobic part of the membrane. Sorry, so it cannot pass through the hydrophobic part of the membrane, okay? So it is hydrophilic. Okay, Ken, next question. Next question is crucial, as in you need to use the information in the passage to outline the process of cell signaling. So before you even do that, you need to remember what you have learned in cell signaling for your syllabus. Outline the process of cell signaling according to your syllabus, but using information in the passage. So first you have to go back to your syllabus. What have you learned about outlining the cell signaling process in the membrane chapter? Okay, so it's this whole thing here. So you need to mention. Now, if it's a 5-mark question, I will expect to have 1, 2, 3, 4, 5. Because this is what you have learned about the steps in cell signaling. So you have to remember this and then only answer the question. So you need to talk about secretion of the hormone by the endocrine gland cells. Okay. And then this hormone diffuse to surrounding and then diffuse into the bloodstream. So number 2, transportation by the bloodstream, circulate throughout the body. Then number 3 is reception, where the hormone binds to the binding site of the receptors on cell surface membrane, or it can be cytoplasm or nucleus for the steroid hormones. But here they say cannot enter, means it must, the hormone cannot enter the cell, means it must bind to the receptor on cell surface membrane. Then followed by transduction. Transduction means conversion of the message, right? So therefore it involves second messenger or intracellular messenger or signal molecule. It activates a series of cascading reactions to amplify the signal. So all these are the important points you have to learn up. Okay, even like the hormone, what is hormone? Hormone is a first messenger or extracellular messenger or the signal molecule. Then what is a response? So it depends on the cell, the target cell. Usually the gene will be expressed. That means transcription will happen, or secretion or movement or metabolic changes will happen, means metabolic reactions. So the question asks you to refer to the passage to outline the cell signaling process. So you need to refer to the passage here to outline the cell signaling process. So step number one, of course, you must mention that the hormone is being secreted. Okay, so chemical, which is the hormone. Okay, so you mentioned that hormone is a signaling molecule, is a first messenger, is a ligand. I'll give you 1 mark. Okay, and then after that, the hormone, so you have to use the information from the passage, right? So some hormones circulating in the blood are able to trigger transcription. That means to say they didn't, the passage did not give us information about what is a cell that secrete the hormone. So therefore you cannot say the endocrine gland cells secrete hormone. There'll be no mark because the passage didn't mention that. So you have to refer to the passage. Even though step 1 is hormones being secreted, step 2, hormones circulate in the bloodstream. Okay, and then step 3, so it's unable to enter the cell, right? So step 3 is therefore the hormone will combine with cell surface receptor 1 mark on the target cell 1 mark. Okay. Some of the students mentioned about G protein. There is no G protein in the passage, therefore there's no mark given because you need to answer the question. So passage has no G protein. Passage also didn't mention about secondary messenger, so there's no mark given. Mark is given if you mention about phosphatases and kinases. So these are the enzymes involved in the enzyme cascade reaction to amplify the signal. So there's mark given if you mention about action of kinases and phosphatases, okay, which is actually cascade enzyme reaction to amplify the signal. Okay, then lastly, the passage talk about response, okay, which is genes being switched on and transcription begin. So therefore, I will expect the student to write the word response. The specific response is transcription or switching on of the gene. So strictly speaking, I want to see the word response to give the mark. Okay, I want to see the word response to give the mark because this is what it is describing. Because if you just copy all gene is switch on transcription, you're just copying from the passage without telling me that this is actually the response. Then I will not give the mark, but even though I've given you mark. Okay, so therefore you have to learn up based on these 5 steps that outline the cell signaling process. That means these are all the keywords: secretion, transportation, reception, transduction, response are all the keywords. So you need to learn up the keywords, right? Transduction or amplifying the signal or enzyme cascade reactions. Okay, so this is for this question. So let's go to the next question. Got any question, please stop me. So use the information in the passage to outline the process, or this one finished already. Okay, next. So you've got amylose and cellulose. They are polysaccharides. So amylose will be like this, right? Whereas cellulose will be straight chain. Okay, so this is one cellulose molecule. So with reference to figure 2.1, state how the structure of cellulose molecule differs from the structure of amylose molecule. So based on the question, which molecule do you need to describe? So I expect you to only tell me about cellulose molecule. This is what the question says. They didn't say state the differences between A and B. So you don't have to tell me about cellulose and amylose. They say, tell me how A differs from B. So just tell me about A will do. So this is how you write minimum and score maximum in exam. See what the question want before you answer. Okay, So therefore, you look at the mark scheme, is only talking about cellulose molecule. So cellulose is a straight chain. If you write, so ignore coil means if you tell me, oh, amylose is coil, ignore, means no mark given, because you need to tell me about cellulose. That's what the question wants. It's a straight chain, it's linear. You cannot say unbranched because both amylose and cellulose are unbranched. Okay. Secondly, cellulose is made up of beta glucose. And because of that, it contains beta 1,4 glycosidic bond. So these two marking points are the same because you are referring to beta glucose. Okay. And then alternate beta glucose are arranged, are rotated 180 degrees. I discovered that some students do not know how to describe this point. So you need to learn up the words to describe it crystal clearly. Okay. Alternate glucose residues are rotated 180 degrees, or neighboring glucose residues are rotated 180 degrees to each other. Okay. AVP, any valid point, that means there are more hydrogen bonds in cellulose molecule because they've got intramolecular hydrogen bonds, right? Compared to the amylose. Okay, amylose. So B, cellulose is the main component of plant structure. Explain why cellulose is suitable as a component of plant structure. So why cellulose is suitable? 4 marks. So let's look at the mark scheme. This is when I see some students have problem to answer this question because certain keywords are not used. That's why you don't get the marks. And some students, they don't really know the structure. That's why they don't give the marks. Some students confuse between, mix up between cellulose and collagen triple helix. Okay. So if you find that you have tendency to mix up, you're not very clear, make sure you study until it's crystal clear before you go for the exam. Okay. So the first marking point that I will give that majority student will score is cellulose has high tensile strength. Okay. So it has high tensile strength. And then second marking point is cellulose molecule is straight or linear. So it's straight. So therefore, it allows the neighboring straight chains to arrange parallel to one another. So because it's straight, it can arrange parallel to one another. And then hydrogen bond can be formed between the cellulose molecules. So this is where students talk about hydrogen bond, but they don't get the mark because they didn't say. You remember when you learn the hydrogen bond, you learn it as intramolecular and intermolecular hydrogen bond. So the word molecule is very important when you describe where is the location of hydrogen bond. So say, oh, there are a lot of hydrogen bonds between cellulose, I will not give you mark. You must say there are a lot of hydrogen bonds between cellulose molecules, or there are intramolecular, intermolecular hydrogen bonds. Because you have got the molecule, you get the mark. So you need to learn up the points crystal clearly. Learn them up, cannot simply change them as you wish. This is called like vocabulary, right? So you have to learn up the word to describe, the word for certain name. You need to learn it up, okay? So when they arrange this way, first they will form microfibrils, right? So we will accept as long as you write fibrils, but strictly speaking, I will not give mark if you write fibrils. I only give mark if you write microfibrils, okay? And a bundle of cellulose molecules will form microfibrils. A bundle of microfibrils will form fibers. Or rather, you write crystal clearly. Okay, why? Because sometimes you see the past year mark scheme can be general in the mark scheme like this. Sometimes will be very strict. So you want to score full marks regardless whether the mark scheme is general or strict. So you always try to give the most accurate, crystal clear answer. Not to pray that they will mark it generally. You don't need to pray, okay? Give the most accurate answer and guarantee you score 90 and above, okay? That should be the attitude you have when you study for bio. Don't pray and hope that the question is easy or the mark scheme is easy. Pray that you can answer all questions regardless the question is easy or hard because you've studied everything. Okay. Okay, so the point number 5 is once the cellulose molecules are arranged in this way, this structure will give strength to the cell wall. So if you say cellulose gives strength to the cell wall, I will not give you mark because the mark scheme already say that, hang on. Because marking 0.4 depends on 3.5 depends on the rest of them. So after you mention this one, then only you say that it provides strength to the cell wall and prevent the cell from bursting. Okay. I think there's one more point. Here. So there's the cellulose fibers, right? They were arranged at angles. That means when the cell wall is formed, it forms in a few layers, right? Where do you learn the formation of cell wall? Chapter 1, right? Okay. Chapter A, when you learn the formation of cell wall. So here, where do I go to? Cell wall here, see? So you see that cell wall are formed in a few layers, right? And then for each layer, so it's like plywood like that. For each layer, the cellulose is arranged parallel to one another. The cellulose fibers are arranged parallel in one layer. And then in the next layer is parallel, but at a different angle to the first layer. So because of the arrangement of cellulose fibers that are at different angle, so it makes it become stronger. So the fibers may run in different direction, like cross ply structure, therefore it's stronger. So this is a marking point for this one here. Okay. Fibers are at angle or crisscross. And then this one, no student mentioned. This is also the function of the cellulose cell wall. That means cell wall is freely permeable. So why is it freely permeable? It's the way the cellulose fibers are arranged to allow many gaps to be formed between the fibers. So that gaps allow passage of water or any other substances, okay, like sucrose. Okay, so that's that. One more mark is given for cellulose is insoluble. Cellulose has got many OH group for formation of hydrogen bonds. Alternate glucose rotates 180 degrees is given only one time, either in 2A, question 2A or 2B. So the same answer will not be given 2 marks in the paper because already given 1 mark. So either in 2A you mention or 2B you mention, once you're given 1 mark, once only. Okay. Okay, let's look at question C. Outline the role of ATP in a leaf cell. 3 marks. So of course, the first mark for the role of ATP is to provide the cell with energy, right? That's function of ATP. Where's my ATP? Okay. So ATP is to provide energy to the cell. And because it's plant cell, the function must relate to the plant cell only. Okay, so any form of anabolic reaction like protein synthesis, photosynthesis, DNA replication, okay, all the biosynthesis, right? So you give 2 examples, you get 2 marks. So 1 mark is to provide energy, 2 more marks is to provide 2 examples. So active transport, okay, proton pump, movement of vesicles, movement of chromosome during mitosis. So this is very detailed. Some students say mitosis. I cannot give mark for mitosis because you must tell me exactly which part of mitosis ATP is used, for example, movement of chromosome. Okay, to that specific process, then only I give mark. If you say growth, of course I cannot give mark because in A level growth, you cannot get a mark. If you say SPM or O level, you can get mark for growth, but A level, to the extent of detail you have learned, to the extent of detail you need to give your answer to score the mark. Cannot give generalized already. Generalized is form 5. Okay. Endocytosis and exocytosis is correct because it's a eukaryotic cell can carry out endocytosis and exocytosis, even though it's plant cell, they can. Okay. Because this is for bulk transport, right? So, any question? So far, so good. Okay, so let's go to question D. Question D got some problem in students' answer. Okay, because the student didn't answer the question and maybe student didn't learn up the meaning of the word. So the question asks, name an example of a source and a sink in the plant. Source and sink. I think some students didn't get this correct. So let's use this to revise the meaning of source and sink. Okay, here. No, not this one. Where's my plant? Plant transport. Source and sink. I didn't open up. Okay, let's go to AS notes. Make sure you get it correct. Okay, translocation. So there is the definition for source and sink here, I think. So make sure you know exactly the meaning of source and sink. Okay, source is any location in the plant where carbohydrate and sugar is formed by photosynthesis or stored in the storage organs. That means to say that when we want to translocate sucrose, where does the sucrose come from? It can come from the photosynthetic organ of the plant, or it can also come from the storage organ of the plant. Like, for example, carrot, okay, storage in the root, or potato is a storage in the tuber, stem tuber, right? Storage organs, okay, can be the source. So it's not only the leaf, it can also be the root or the stem. Okay, so leaf, potato tuber, onion, carrot root, they are all the source. Okay, sink are the location in the plant anywhere that the sugar is either being used, okay, so it can be used for respiration to produce ATP, it can be used to produce cell structure, okay, in growth, like for example, phospholipid molecule, protein, okay, chromosome, lipids, and so on, produce organelles, okay, or to be stored. So this is called the sink. So make sure you know what is source and sink. So when you learn biology, learn up the whole meaning. Don't learn half-half. So you need to allocate a lot of time for you to learn up all the details for biology. You need to allocate the time and you need to learn them up, okay? No last minute work, okay? Because a lot of details to learn. Name cells C and D. So very straightforward. Cells C and D are C is the sieve element or sieve tube element. D is companion cell. Okay. Part 3. Explain how sucrose travels from the source to cell C. Okay, from the source to cell C, 3 marks. This is where a lot of students write unnecessary answer wasting time, right? Because when you spend time writing answers that already the question didn't ask, you're wasting your own time in exam. So you got less time to answer other questions. So don't do that. Answer only what the question asks for. So you only need to explain the mechanism how sucrose from the source, which is here, travels to cell C, which is this one only. From here to here only. Don't tell me about the translocation in the sieve tube from the source to the sink, because the question didn't ask that. So wasting time only writing all this answer. Okay, so let's look at the mark scheme. Very straightforward, right? Proton is actively pumped out from the companion cell to the mesophyll cell wall. To the mesophyll cell wall. So that means to say that it is being pumped, okay, from this companion cell, pump out the H plus from the cell into the membrane, sorry, not membrane, through the membrane to the cellulose wall of mesophyll cell. So this is mesophyll cell. Meso means middle, phyll means leaf. Middle of the leaf. Cell in the middle of the leaf means the cells sandwiched by the upper and lower epidermis in the leaf. It means palisade and spongy mesophyll cells. Okay, so to the cell wall of the mesophyll cell. Next, sucrose will then move into the companion cell through co-transporter or any other word with the same meaning. It means sucrose co-transported with proton into the cell, 1 mark. Next is sucrose will then diffuse down the concentration gradient into the sieve tube element. That means from the companion cell, sucrose will diffuse into the sieve tube element, down concentration gradient. Okay, down concentration gradient. Then after that, through plasmodesmata is 1 mark. Okay, through the plasmodesmata. So that's all the answer. Nothing else already. Because you don't have to talk about water entering, okay, then what hydrostatic pressure increase and pushes the sucrose to move from source to sink. No need, because that's not what the question asks. No need to write everything. Okay, read question carefully, see what the question asks for, give only the part that the question asks for, right? Save your time to answer other questions. Okay or not? So shall we continue with this question? So this question, they say that they have prepared 6 reaction mixture, and these are the composition in the reaction mixture, right? So they're talking about the urease activity, okay, which convert urea to ammonia. Okay, I think plus CO2. Okay, so this is a reaction and catalyzed by the enzyme urease. So it's good that you actually write this every time when it's enzyme catalyzed reaction, write the equation, write the enzyme, so you know it clearly. So ammonia is basic, so it will actually increase the pH of the reaction mixture when ammonia is formed. Okay, so they said there are 2 chemical inhibitors that are given. So thiourea and lead nitrate could be the inhibitor. Could be only. So we need to look at the question first. Okay. So let's look at the question. So they say that the student recorded an increase in pH in reaction mixture 1 and 2. So increase in pH means ammonia is produced, right? That's why it's basic, so the pH increases. Okay. The reaction was faster in 1 than in 2. That means 1 and 2 got reaction, but reaction is faster in 1 and 2. Okay, so the question asks a student. So I'm going to clear everything with you. So the student made conclusion, match the statement to reaction mixture. Number 1, no reaction took place because urease was denatured. So this is very easy. You boil the urease, its denature is number 6. So part 1 is number 6. Okay, part 2, there was no reaction. So this is where a lot of students didn't get it correct. No reaction. Sorry, this one is okay. Not this one. Because there was no substrate. Still, this is quite easy. So just look for the reaction mixture without urea is a substrate. So 5 got no urea. So answer is 5. The last one is the one that a bit challenging. The reaction did not occur. That means no reaction. So the statement earlier already mentioned that 1 and 2 has reaction. So that means 1 and 2 increase in pH means got reaction, right? So which one got no reaction then? Then you see, because of inhibitor, there are 2 possible inhibitor, which is thiourea and lead nitrate. This one got no urea, so therefore there's no reaction because there's no substrate. So therefore this is not the answer. Then this one is also not the answer because there is reaction. So the only possible answer is this one here. No reaction because lead nitrate is the inhibitor that inhibit the reaction. So answer is 3. So this is where a lot of students didn't get the answer correct. Okay, this is 3. Okay, so here there's a graph given. What you need to do is that you need to sketch a curve on the graph to show the results that the student would expect if thiourea act as a competitive inhibitor. So this is very straightforward. So most students get this answer correct, majority. Okay, so if it's competitive inhibitor, you start with the curve lower than the original, right? Because you got competitive inhibition, but eventually it will have to reach the maximum. Okay, so 1 mark given for starting from 0 and the curve lower than the original curve without inhibitor, and finally end up at the same maximum rate of reaction as this curve. So 2 marks. So most students get it correct, right? So let's finish the last answer. Explain why is it important to determine the initial rate of reaction when investigating the effect of competitive inhibitor on enzyme. So whether you're investigating competitive inhibitor or not, as long as you want to measure the rate of enzyme catalyzed reaction, you will always have to measure the initial rate, right? Because you know that the substrate will be converted to product whenever enzyme is mixed with the substrate. So therefore, once the reaction starts, your substrate concentration will actually drop already. So when the substrate concentration drops, that means to say that the rate of reaction will also drop already. Okay, so therefore, you want to measure the initial rate because the initial rate is the highest rate. And this is the rate that is closest to the initial substrate concentration that you have reported in your materials and method. Therefore, the rate is closest. So therefore, you have to measure the initial rate. Okay, do you see? Because once the reaction starts, the substrate is going to convert to product, its concentration is going to drop. So when the concentration drop, the rate will drop. So therefore, the highest rate is right at the beginning, which is called the initial rate, when the substrate concentration is the highest and is closest to the reported substrate concentration that you use. So that will be the most accurate rate that you need to measure. Okay. Other than that, whenever you got inhibitor, now the ratio of inhibitor versus substrate. Of course, hang on, I put it the other way around. The ratio of substrate to inhibitor is the highest at the beginning. Highest at the beginning. Because we're talking about competitive inhibitor. Competitive inhibitor means my inhibitor will compete with substrate for the enzyme active site. So at the beginning, substrate concentration is the highest, isn't it? Highest S to I ratio. Therefore, there is higher chance for substrate to sit into the enzyme active site. So therefore, the rate of reaction is the highest. So when they ask you about measuring the rate of reaction where it involves competitive inhibitor, you need to say that the ratio of substrate to inhibitor is the highest at the beginning of reaction. Therefore, the rate of reaction is the highest because there's less inhibitor that can sit in. There's more substrate that can sit into the enzyme active site. So the rate of reaction is the highest at the beginning. Okay. Lastly, you do not measure at the end of the reaction because at the end of reaction, there's no more reaction. So the rate is actually equals to 0. Right? The rate is the gradient of the curve is 0. So you don't measure any reaction at the end of reaction because the rate is the same as 0. All substrate converted to product. Okay? Ken, so you want to see the mark scheme? I already explained the answer for the mark scheme. Okay? So initial rate is the fastest because it's a higher substrate to inhibitor ratio. So the enzyme is most active. Least inhibitor can fit to enzyme active site because you've got more substrate to inhibitor. At the end point, the rate of reaction is 0. So no point measuring at the end. All will be the same. So there's a maximum difference in the rate among different reaction mixture at the initial, at the beginning only. At the end, all are 0, right? So when is it the most different among the different reaction mixture is at the beginning. And then the substrate concentration, inhibitor concentration are as indicated in materials and methods at the start of the reaction. This is referring to substrate concentrations at the start is nearest to the reported substrate concentration in the materials and methods. Okay, so that's all for today's discussion. We'll continue with the discussion in the next lesson. All right. Okay, thank you, everyone.